Hi everybody, welcome back to Bio 21. This week we'll be talking, or this lecture I'll be talking about the cell cycle, mitosis, and meiosis. So let's go ahead and get into the material. If you're ever trying to figure out where I've been or where I'm going, here's our lecture outline. We'll be covering the three things I just stated, so the cell cycle, mitosis, and then meiosis, but we'll also spend a little bit of time talking about chromosomes, their structure and their number, and other cool fun facts about them. Okay, so let's jump into the cell cycle. I love this slide. Oh, so cool. I wish it was real. Remember, think back to cell theory. Remember that the cell is the smallest unit of life that we have. And so every single cell that your body has came from another existing cell, right? That was the first rule of the cell theory. I think it's important to just take into account the fact that our cell cycle is what our cell is doing when it's not dividing. So we'll talk about the stages, but essentially we thought that cells just kind of existed for the purpose of dividing and making more cells in multicellular organisms. And then when we started to really look at what the cells were doing in between splitting events, we saw that there are two major stages and that there's a lot of stuff that your cell's busy doing when it's not dividing. Right? Think back to our signaling transduction pathways lecture and our cell membrane communications when we were looking at organelles. Right? Our cells are highly regulated and they're doing lots of jobs at different times, and these events are sequential. They have to be sequential. And they have to be choreographed and perfectly timed. None of that has nothing to do with cell division, right? And so there's a lot. Then we talked about cells that we haven't talked about in Bio 10. We've talked about cells that don't do much division, right? They're cells and tissues that if you have damage to them, it doesn't heal back, or it takes a long time to heal back, or it doesn't heal back the exact way it was before the injury, right? Those types of tissues don't undergo cell division very often, and it takes a lot to get your cells to divide sometimes too. So just take that into account when you're thinking about, as we go through these next couple slides talking about the cell cycle. We're going to be talking about what your cell's doing when it's not dividing. Okay. When it does divide, it'll do it in one of two ways, right? It'll either be for mitotic division or meiosis division, and this is specifically for eukaryotes. With our prokaryotes, it's a different ball game. But you can either make more cells so that the organism can have get bigger and have more tissue differentiation, or you can make more cells because you want to reproduce the individual or pass on their genetic information to a completely different individual, and that's the difference between mitosis and meiosis. So we'll talk about that as well. So for the stages of the cell cycle, these are them. We'll talk about them each over the next couple slides. So again, cells are the simplest units of life, and when your cell is after it has split, right, after that first division event, and your cell is a brand new cell, it's going to be growing. It's going to be growing larger, and this is going to happen primarily during the G1 phase. So we have a G1 phase and an S phase and a G2 phase and the M phase. We, like I said before, thought cells only kind of got bigger and then divided, and we thought that was the point. That was it. And so we originally had meiosis or mitosis, and then we had like a stage where you duplicate the genetic information in the cell. And we thought those are the two major events. So we have the M phase for cell division, and then we have the S phase for synthesis of the DNA. And we didn't really pay much attention to G1 and G2 in the beginning. we now do, so that's why they have names, but they're called gaps. They're called gap 1 and gap 2. And that's why they're called gaps, because the focus was on mitosis at first, and DNA synthesis or replication. And then there were just 2 gaps in between those 2 periods of time. Okay. So during gap 1, during the G phase, your cell is getting larger in size, and it's synthesizing a lot of molecules that are involved in that growth and not differentiation. I won't say that. I'll reserve that for multicellular things. But it was G1 is dedicated to signaling molecules that trigger cell growth and cell maintenance. The S phase is all about DNA synthesis. And so this is the stage when we are doing chromosome replication. And we will talk about chromosomes at the end of these slides, but there's a lot of jargon that's important for you to learn so that you can be clear on what structure we're talking about at a given moment in time, especially when we're talking about meiosis. So you have 46 chromosomes in your body. You have 23 chromosome pairs, and for every chromosomal position, so chromosome 1 has 2 chromosomes, sister chromosomes. We call them sister chromatids. They're chromatid and chromosome are interchangeable names. Really what you're talking about is the thing that has a centromere. And so sometimes your centromere has 1 sister chromatid attached to it. Sometimes your centromere has 2 sister chromatids attached to it. Either way, we call that a chromosome, and I'll show you this on a slide at the end. But when you read in this statement here that there's a replication of the chromosomes and each copy is called a sister chromatid, I just want to define that for you a little bit more. So, okay, and I think everything I just said is here. The human cell has 46 chromosomes. And so after the synthesis phase, you will have a duplication of those chromosomes. So you'll have 46 pairs, and each pair is made up of 2 chromatids. And we call them sister chromatids because they carry the same genetic information on them. And we want to keep them organized in those 46 pairs. You don't want to put your X chromosome with your chromosome that codes for your eye color. They're not going to pair up properly during DNA translation. What else? Okay, that's it for this slide. Okay, for the G2 phase, it's preparating for mitosis and cytokinesis, right? So there's going to be a lot of proteins that are involved in think of it as a ballet. The chromosomes are kind of moving around and bumping and remaining in their pairs, and they have to do all of this in the cell at the same time. So there's a lot of proteins that have to be synthesized in order to coordinate this movement. And then there's proteins involved in the division, the ripping apart of your cell into 2 different new cells. That's called cytokinesis, and that requires its own set of proteins. So G2 is dedicated to creating those proteins and getting them in the right position in the cell. And then it's go time. When we hit mitosis, everything is kind of already in motion, and it's too late to stop the train. So you want to make sure that everything was done properly in the preceding phases. And mitosis, we'll talk about the stages of that, but that's all about your cell, dividing its nucleus into 2 and separating out all of those sister chromatids so that each daughter cell has the same genetic information as the parent cell. Cytokinesis is simply the splitting up of the cytoplasm. So that part is kind of its own. In terms of timing, it's important to realize your cell has a lifespan, right? Each cell has a lifespan just like you and I, and they spend time in different parts of their lifespan, different amounts of time. For example, you spend so many years as a child, you'll spend so many years as an adult, and you'll spend so many years as an elderly person, and so as an elderly adult. And we represent the phases of the cell like we do those age groups. So the amount of time that your cell spends in each of its phases depends on the type of cell. Some cells are constantly dividing, and so they're going to spend a lot of their time in M phase, but that's rare. Most of our cells aren't doing that throughout their lifespan. They actually if their lifespan was in 24 hours, they would be spending most of their day in G1. And they would spend about 8 hours just carefully synthesizing all of that genetic information, all that genetic material in preparation for cell division. And they only spend 4 hours in G2, making all the proteins that are needed for mitosis to occur. And then mitosis itself all happens within an hour. If you have the whole day, think about your whole day and think about how fast an hour flies by. Right? It's like pretty fast. So cells are not spending much of their lifespan dividing, most cells. But this is all very much broad strokes, guys, because I'm talking about a generic cell. And as you know, there's lots of different types of cells. We're going to talk about lots of different types of tissues, and so that you can keep this in mind as we're going through. Think about how these lengths of time might change depending on the type of tissue you're looking at and the type of cell you're looking at. The decision to divide is complicated. It entails both external factors and internal factors. So there are things that tell the cell it's time to divide outside of the cell's control. There are molecules that are sent from like your pituitary glands and the other parts of your body that signal cell division in other parts of distantly related parts of the body down in your reproductive organs, right? So that is what goes into whether or not a cell divides. And then there's what's happening inside of the cell itself that goes into whether or not it divides. So there are cell cycle control molecules. We'll talk about those in a second. And there are checkpoints that the cell has to progress past in order to undergo cell division. If it doesn't progress past those checkpoints, it doesn't divide, right? And we talked about that when we were talking about cancer and talking about gene regulation. So yeah, it's all coming together.